Imagine a young girl browsing the doll aisle of a toy store. She notices the huge selection of dolls that the store is offering, all so different and all so unique. She likes the hair of one doll, but also likes the eyes of another. Luckily, the store allows her to personalize her doll and choose the particular characteristics she would want, such as hair color, hair texture, facial structure, and eye color. Now imagine applying this concept to humans. This is almost possible through the process called gene editing. All of the material that makes us unique is encoded in our double-stranded DNA sequence found within our cells. Gene editing is a phenomenon where DNA, our biological blueprint, can be modified. DNA is composed of numerous genes which dictate our physical characteristics, as well as the diseases we may be susceptible to. These genes are made up of smaller building blocks called nucleotides. The entirety of our DNA sequence is composed of 4 nucleotide bases: adenine, thymine, guanine, and cytosine. So, how does the process of gene editing work? You may have heard about the famous gene editing technology called CRISPR. This technology relies on a complex called the Cas9 complex. This complex consists of 2 main components, a Cas9 protein and an mRNA strand. The single-stranded mRNA is able to guide the Cas9 complex to the target area on the DNA sequence. When bound, the Cas9 protein is able to cut the DNA at specific locations, and this allows for the insertion or deletion of nucleotides. To understand this better, imagine this railroad track represents DNA and this train represents the Cas9 complex. mRNA can be thought of as the train engine that leads the train across the DNA. The Cas9 protein is comparable to a repair wagon on the train that removes and modifies sections of the track. By changing nucleotide bases, this in turn alters the product of the gene. What this means is that an individual that once had a disease-causing genetic sequence could use CRISPR technology to remove or edit this harmful DNA. The opportunities that gene editing provides are endless. It could be applied in medical settings to allow for the treatment of human diseases, as well as in agricultural context by genetically modifying the traits of particular crops and animals.